in some genes transcribed by RNA polymerase II, a sequence of nucleotides, that's a cutter box that is nearly identical to that of the plasma box, which is present in the prokaryotic organism, which is standard about 35 upstream to transcription. So this was as identical to the plasma box, which is a prokaryotic organism. In a eukaryotic organism, there is minus 25 upstream cutter box, and the name of the cutter box is Hawkins box. So the co-promoter sequence is called a Tata box or Hawkins box. In majority of the genes, however, there is no Tata box is present. Instead, different co-promoters such as initiators or DPE, downstream promoter element, they are present instead of a Tata box. They all can be present simultaneously, they can be present separately, and for the initiator or process of transcription. Now they are coming to another topic, that is general transcription factors. We have already discussed the general transcription factors which are necessary for the process of transcription besides these RNA polymerase enzyme. So this is a DNA molecule having an RNA polymerase II, which is helping us for synthesis of messenger RNA. These are transcription factors. Transcription factors are named as transcription factor 2. They can be transcription factor 3. They can be transcription factor 1. This subunit, which is E, B, D, H, and F, they have another individual functions which can be demonstrated separately. The most important is a D1. So they have TF, transcription factor 2D, and GFT, general transcription factors contain a Tata binding protein. So the D is important. Why D is important? Because we have a binding protein. What's binding protein? Tata binding proteins. Because TF2 is the first thing which gets itself attached to the Tata box. This is the Tata box. So transcription factor 2 gets attached itself first. This attachment of transcription factor 2 will bring RNA polymerase II. If there is no transcription factor 2D, there will be no bringing up of the RNA polymerases. Transcription factor F is another general transcription factor with the polymerase, which will bring the polymerase to the promoter region. The helicases are here from H. You can remember the helicases activity, which melts the DNA, and its kinase activity, phosphorylase polymerase enzyme, allowing it to clear the promoter, and then this promoter is removed. And this is a eukaryotic area. This is a Tata box. This Tata box, and this is point of replication. This Tata box, attachment of transcription factor 2D, it attaches itself to the Tata box. How it gets attached? Because it contains a special sort of proteins. The name of those protein is TPP, binding proteins, transcription factors binding protein. So these transcription binding proteins get attached itself to the Tata box, and it will bring different transcription factors. They are transcription factor H and they are transcription factor F. Transcription factor F will bring the polymerase, and transcription factor H will match the DNA, and it has the activity of helicases activity, so it will match the DNA. This is another enzyme. The gene is turned on. Why the gene is turned on? This gene, transcription, messenger RNA, and translation. By now, we must know the process of synthesis of messenger RNA is helped by an enzyme. The name of that enzyme is RNA polymerase II. So this RNA polymerase get attached itself. This Tata box, transcription factor 2D. Transcription factor 2D has special sort of proteins, TPP, transcription binding proteins. Transcription factor 2F, what will you have to do? It will bring RNA polymerases. Transcription factor 2H, it will helicases, unwind the DNA, and unwinding the DNA and remove the promoter area. Coming to another very important stuff, that was the steroid hormone. Steroid hormone, being steroid in nature, it has the ability to cross a cell membrane and cross the cell membrane and get attached itself to a receptor. Binding of steroid hormone to a receptor. This is a receptor, the yellow in color. This hormone attaches itself to the receptors and moves inside the DNA. When it moves inside the DNA, it gets itself attached to DNA and binding of the complex to the DNA regulatory site. The DNA regulatory site is expectively a chatterbox in a eukaryotic organism. This DNA then will help us in the process of transcription. What is transcription? The synthesis of messenger RNA. This messenger RNA then goes to the ribosomes and the process of translation is there and new protein is synthesized which is acting